For part 2 of chapter 9, we're going to start with the central dogma of biology. The central dogma states that that DNA is turned into RNA, and then RNA is then turned into protein. We now know that this actually isn't true necessarily. There are viruses that are made out of RNA, and they turn themselves into DNA, or they're encoded into the host's DNA, and then that DNA is turned into RNA, which is turned into a functional protein. So you follow this pattern, but you can't actually work this pattern backwards. But that's the second part of this chapter, is how. How do you go from DNA to RNA to protein? And that's what we're going to look at. So going from DNA to RNA is referred to as transcription, because you're transcribing a message, you're writing out a message using the exact same language that you used previously. The first thing you have to do in order to transcribe a message is to initiate the process. And to do that, the DNA double helix must unwind just a little bit at the region where you would like to make your messenger RNA. The little bit that you unwind, we call that the transcription bubble. We're going to see we use a lot of the same terminology. There are a lot of different proteins and enzymes that are involved in this process, but what they look at first when they go to create that transcription bubble is a little tiny recognizable sequence of DNA referred to as a promoter. Typically speaking, that exists upstream, or if you're reading the DNA like a sentence, it would exist, a few sentences beforehand, a few sentences, higher on the page. We call that upstream. So we're going to initiate this process by finding the promoter with very specific enzymes and proteins. And it is going to dictate what happens. And this image kind of shows you what happens. So you open up your DNA that's been located appropriately because of the promoter, and you create a little bubble, and an enzyme known as RNA polymerase is going to come in, just like DNA polymerase did, and link RNA nucleotides that are perfectly complementary to the DNA parent strand. You run your polymerase down until you hit a stop point in your DNA. The RNA polymerase releases, DNA ligase comes in and seals your DNA back up, and you're good to go. And then you can start altering that RNA to make sure it's exactly what you want it to be. Here's a little bit more information on that elongation process. Transcription always proceeds from just one of the two DNA strands. We give it the moniker the template strand. The RNA product is complementary, so it matches up. You have that same pairing where cytosine always pairs with guanine, but in this instance, when you would like to lay down a thymine, you can't. You have to lay down a uracil as well. So it's A and U rather than A and T. The RNase polymerase proceeds along the DNA template, just adding your nucleotides one by one. The RNA doesn't remain, stuck to your template. It kind of hangs out on the end. As elongation proceeds, the DNA is just continuously unwound, just like when you move a zipper, and then rewound right behind it as you move along. We don't leave it open for long, so you can kind of see how this process is running. And again, we do it in the 5' to 3' direction. So you add your 5' first and build to your 3'. Just like before, it's very similar to a process of DNA replication. So once you transcribe a gene in prokaryotes, a prokaryote polymerase needs to be instructed to dissociate from the DNA template. So what that means is that you have to have special instructions to let the polymerase, like, hey, it's time to let go. There are a couple different signals that let the polymerase know that. There are two different kinds. They both involve really repetitive nucleotide sequences, and they result in the RNA polymerase stalling. So it physically stops moving on the DNA strand, and the protein lets go because it essentially gets jammed with information. In a prokaryotic cell, when termination has occurred, there's a really good chance that you've actually already synthesized your protein. You've actually already turned your RNA into proteins, because there isn't anything separating the DNA from the rest of the cell. Turning RNA into proteins is called translation, and translation happens immediately thanks to ribosomes that exist in that nucleoid region. And we're going to take a look at what that means, and it's a little different in a eukaryotic cell. Well, it's a lot different in a eukaryotic cell, I should say. So this is what I mean in a prokaryotic example. So you have the DNA and you have multiple RNAs that are being made, And attached to those RNAs are just a ton of ribosomes. And this is one of the reasons that bacterial genes can be copied so quickly and why bacteria are capable of growing so fast and doing what they need to do as fast as they can, because there's no separation between this process of going from DNA to RNA and then RNA to protein. It happens almost simultaneously. It is, of course, different in a eukaryotic cell. So when we look at a eukaryotic cell, we end up doing a lot of grooming to our mRNA that we very first make, and that's what you see up top, is the primary RNA transcript. That's the transcript that first comes off of your DNA, and it turns out we have to do a lot of work to kind of make it ready to go to be transferred into the cytoplasm for RNA processing. So you can see they've labeled a few different things. They have exons up there and then introns. It turns out that large parts of our genes don't actually code for a practical protein product. They're just called intervening sequences. That's what that intron stands for, intervening sequences. And what it means is that we can cut out those little bits, those little introns that don't code for anything. And if we would like, we can reshuffle all of the exons or the sequence that will be able to exit the nucleus in order to do work. So if you kind of like, I don't understand the purpose of that, think about how many words you would be able to write if you always had to use the alphabet in order. You always had to write your letters A, B, C, D. You couldn't shuffle them at all. It would really restrict the number of words that you're capable of writing. However, you can move them around and you don't have to use all of them. And it turns out we can do that with our exons, with our little pieces of DNA that actually code for protein. And that allows us to make many, many different kinds of protein from one little strand of DNA, just like we can make many, many different words from one little alphabet of only 26 letters. So the process of cutting out the introns and splicing the exons back together in whatever order we need them in is known as the alternative splicing of RNA, and it leads to kind of a more advanced processing of our genomics. After we splice our RNA, the next thing that we need to do is add a 5' cap. The 5' cap is made up of a whole bunch of repeating guanines. It's just a really long strip of guanines, and it allows the RNA to be essentially tagged with a front end and a back end. On the back end, we add a whole bunch of adenines. We refer to it as the poly A tail. When I say a whole bunch, I mean about 200 or 250, depending on the RNA that we're looking at. But this nice RNA now that has undergone splicing and has both its cap and its tail, and its little untranslated regions that just allow it to be essentially threaded through the ribosome like a thread through a needle. Those exist as well. Then this little fella is allowed to go out into the cytoplasm in order to be copied and turned into a protein. We do that in this really big complex known as a ribosome. Protein synthesis machinery includes a large and a small subunit of the ribosome. We, of course, need the help of our nice mRNA that we just created, as well as a tRNA, which is that big yellow structure with the large growing polypeptide out the top of it. So those little tRNAs actually have a pretty interesting structure and therefore a pretty interesting function. You'll notice the little prong end. That little prong end is known as the anticodon end. And the anticodon end looks for really specific 3 nucleotide chains, because the three nucleotides in a row are a code for a single amino acid. 3 nucleotides equals 1 amino acid. And you'll remember, hopefully, that amino acids are the building blocks of proteins. So it's by pairing the tRNAs with the mRNAs that you end up building your protein. That's where the instructions lie, in this little 3 amino acid code. And we'll see the code in just a second. But how this whole kind of situation works is the ribosome holds the mRNA and the tRNA essentially like in the appropriate position, and then when you line up a few tRNAs next to one another, they're a little amino acids that naturally are attached to the end of them leave the tRNA and they bond with one another. Then your empty tRNA, it leaves, and the whole little unit kind of hops over one, and you can add another tRNA that's holding on to an amino acid, and you just keep doing that. You form a bond, you leave. You form a bond, you leave. And that's how protein synthesis works. That's how this machinery actually works. So it's kind of a neat little setup. It's kind of ingenious, if I do say so. So let's take a look at that actual amino code. So here's what the code looks like. This figure shows you the genetic code for translating each nucleotide triplet or codon of mRNA into an amino acid. We refer to this process as translation because we said before that DNA to RNA was transcription because you're writing a message in the same language. Well, we switched languages. Now we're going from the language of a nucleotide into the language of an amino acid, so we kind of have to, use a translation chart. We have to figure out what our words mean. And we have done that. Scientists figured that out in the 70s and the 80s. So here you go. Here's the little chart. What this shows us is the first letter of the triplet code, the second letter, and then the third letter. And you kind of just use this to narrow down what you're looking at. It's a little bit like using a map. If you've ever used a map and you've used the key code, the A and the letters on the side and the numbers on the top. So say maybe we are looking for the amino acid AAG. We're curious what adenine, adenine, guanine makes. Let's figure out what row we're working with first by considering just the A row. And then we go to the top and we consider just the A column. And then we go to the side and we consider just the skinny G row. And we see that AAG codes for the amino acid lysine. And you can actually go through a whole sequence of mRNA if you'd like and figure out what the resulting protein code is. It's kind of neat. There are a few on here you might notice that are different colors. They're standing out. There's one codon that always means, hey, start right here. AUG means start right here. Every single amino acid, every single mRNA that we know of that codes for a functioning protein starts with AUG. There are 3 different codes that mean, all right, stop right here, we're done. And those you see up top, the AUU, UAG, and UGA. You might notice there's some duplications here. There are 4 different ways to say arginine. There are 4 different ways to say glycine. 4 different ways to say valine. What's going on? This is called redundancies in this program, and they're actually kind of important. So those amino acids tend to have very important bonding structures. And what that means is if you do actually make a mistake, you were supposed to say CGC in your mRNA, but you accidentally made a mistake and you put CGU. If you look at the arginine box, that's fine. You're actually still going to end up with the same amino acid. It's kind of like having a backup. It's autocorrect for genetic processing, but instead of autocorrecting what the word says, the outcome is still just the same. So that's kind of a neat feature of this process of DNA to RNA to protein transcription and translation. There is some autocorrecting in there. So here's that little process again, because it sounds a little funky. Here's the process of translation. It starts when a tRNA with the right anticodon end, you can see how they match up in this picture, recognizes the appropriate start on an mRNA. The mRNA right now only has the bottom of the ribosome, which to me looks like a hamburger bun, attached to it. Once you get your first tRNA, the larger ribosomal subunit, the top of your hamburger bun, will come in and sit on top of it and kind of bind everything together to ensure that everybody's nice and close for this process. Then a second subunit of tRNA is going to come along, and you'll see it's carrying its little amino acid. What happens is the mRNA moves relative to the ribosome, so it kind of pulls through the ribosome, and as these little amino acids get close to one another, the tRNA helps them to bind, and you form a nice long polypeptide chain. Once you give up your amino acid, you leave, and then everybody scoots over a chair. Someone new comes in. The first chair gives up its amino acid, it leaves, and everybody scoots over a chair. And you just keep going. You keep going back and forth, back and forth, back and forth. We have specific sites for all of these. You'll notice that they're labeled E, P, and A. The E, so whoever's sitting in that very first chair, he's the one that's getting ready to exit. P is your primary, and then A is that third site. But you just toggle back and forth, and this is how you make your protein. When it comes to eukaryotic gene expression, it's really regulated during transcription and RNA processing. It's not at all like that prokaryotic process we just looked at. So a lot of this processing, or excuse me, this genetic expression takes place inside of your nucleus, as well as the protein itself during translation, so it takes place inside of the cytoplasm. So before we saw it all happening in one little spot right next to the DNA. Definitely not the case in higher order organisms or eukaryotic organisms. It can happen a little bit of everywhere, and a lot of it takes place in the nucleus, but there's plenty that can occur in the cytoplasm also. There are 5 different kind of models or modes for that alternative splicing that we discussed, that we talked about. And you can kind of see those different modes. We don't completely reshuffle all of our exons. It's not like, just throwing them in the grab bag and picking out random ones. Usually, there's some like really basic options. You can just skip an exon. Some certain exons can be mutually exclusive of one another. Sometimes you just kind of flip the 5' site, sometimes you flip the 3' site, and then there are certain instances where you actually keep your intron. So it gets a little funky. There's a lot of things you can do to create some pretty cool genes. But with that, it's actually the end of our discussion of RNA translation. You can get kind of crazy with this stuff, and there's some really, really cool topics out there, but they're not quite the scope for Bio 100, but they're pretty neat all the same. So you should look up what you can do with alternative gene splicing or expression, because that's kind of where the science is headed. With that, you should take a look at your book, consider all of your possible study methods. There are some wonderful animations and videos out there for these processes, and it's really helpful to watch them happen before your eyes. You should listen to multiple resources and consider everything that's available to you to study. It's always a great idea to listen to multiple resources. And have a great day.